All right, moving on to lab 2, we're going to be talking about genetics and inheritance, specifically about patterns and mechanisms. Some key terms. If you guys have ever taken a high school biology class, then you're probably somewhat familiar with Punnett squares. But Punnett squares are a method of diagramming inheritance where parent genotypes are used to estimate the probability of potential offspring genotypes. Now, an allele is an alternate version of a gene, and these can be dominant, which will always be indicated with an uppercase letter. And dominant alleles mask the effects of other alleles for a trait, or they can be recessive. In this case, it will always be indicated with a lowercase letter. And these are going to be the alleles that are being masked by the dominant allele. A gene is just a section of DNA that codes for a trait. A genotype is the specific alleles that an organism has for a trait. So it's going to be the specific combination of letters that is going to represent that trait. A phenotype, on the other hand, is the physical expression of an organism's genotype. So it's what we can actually visually see. Now, genotypes can be homozygous, homo meaning one. They can be homozygous dominant, so they have two dominant alleles for a trait. In this case, we're using B. We're going to have two capital Bs. They could also be homozygous recessive. And in this instance, we're going to have two recessive alleles, so two lowercase b's. They can also be heterozygous, though, so they're going to have one dominant and one recessive allele. And this is going to show up as one uppercase letter and one lowercase letter. Now, here in the photo, let's visualize that these are flowers. So this dominant or capital B is going to code for red, and this lowercase or recessive b is going to code for blue. Both of our parents in this instance are heterozygous. And when we finish our Punnett square to kind of get our potential offspring, you can see that we have one homozygous dominant offspring, two heterozygous offspring, and one homozygous recessive offspring. What this means for your phenotypes is that one dominant or homozygous dominant offspring is going to be red. There is no recessive alleles present in this offspring's genotype. The heterozygous offspring, you can see we have two of these. Despite having one of each allele, because we are talking about complete dominance, that dominant allele or capital B is going to overtake the recessive or lowercase b. So these are still visually going to look red. Moving on to our one homozygous recessive, because there is no dominant allele in this genotype to overtake the recessive, is going to be the only offspring that appears blue. And we're pulling all of this from Mendelian genetics, which we talked about in the last lecture. And it uses kind of this blending model, and it's based on Mendel's theory of particulate inheritance. Now, he uses true breeding pairs. This is very important because it helps to avoid interference from other forms of inheritance or other types of inheritance, which we will talk about in the next few slides. And what this means is that he is using one parent that is homozygous dominant and one parent that is homozygous recessive for the first breeding pair. This first breeding pair is actually called the P generation or the parent generation. Their offspring is going to be all tall. If we fill out the Punnett square for that, you will see. We'll go over that in the next slide. But they're all actually going to be heterozygous. And that F1 just means first filial. It just means that it's the first offspring generation. Then we move on to the F2 or second filial generation. This is the second generation of offspring, based on the idea that their parents are going to come from that F1 generation. So because the F1 generation in this case is all heterozygous, both the parents for this F2 generation are going to be heterozygous, and it's going to result in 3 tall plants and 1 short. Here you can see this as well. This will be very helpful for your guys' homework. But the P generation, we have that homozygous dominant parent and that homozygous recessive. And you can see that we are placing 1 parent across the top and 1 parent down the side of our Punnett square. Each box gets 1 allele for the parents. And when we fill out those boxes for the offspring, they're going to get 2 alleles, so they're going to pull 1 from each parent. You can see that in this case, they are all going to be heterozygous, which means that their phenotype is going to be all tall, although you really should put these in a ratio format. In this instance, it would be 1.0 because they are all tall. Don't put 4.0 or 4.0 because we need to simplify. Moving on to the F2 generation, we're going to steal 2 of the heterozygous offspring from the F1 generation to be our parents. So they're both going to be heterozygous. And when we pull those down and fill out our Punnett square, we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive offspring. The phenotype for this, or phenotypic ratio for this, is going to be 3 tall and 1 short, because we lump in those heterozygous with the homozygous dominant, because they all look visually tall. Now, the other forms of inheritance that we were talking about that Mendel wants to avoid with his use of true breeding pairs, one of these is incomplete dominance, and this is when a dominant allele can't completely mask the effects of a recessive allele. This results in a blending of the alleles in the offspring. So it creates a third phenotype where it is a blend of the 2 parent alleles. In this instance, we can see with this flower that we have a homozygous dominant parent that is red. Red is going to be the dominant color in this instance. And then 1 parent that is homozygous dominant for white. When they combine, because they can't completely mask each other, they're going to blend to form pink offspring. Now, this only works when the offspring are heterozygous, of course. Otherwise, complete dominance would take in effect. There's also codominance, and this is when neither allele can mask the expression of the other allele. And as a result, the phenotype of the offspring is a combination of the parents. So it is a combination, but it's not a blend like incomplete dominance is. Here you can see in the photo that we have a blue and a white bird, and their offspring, because these alleles are fighting with each other, they're going to combine instead of blend. So our offspring is going to have a blue body, but a white stomach. If we go back to this flower kind of analogy for codominance, the flower, instead of being pink for the offspring, would end up being white with red splotches or polka dots or streaks, or vice versa, and it'd be red with white polka dots and splotches and streaks. Now we are going to go over these Punnett squares a little bit together, just so that I feel that you guys are fully prepared and know the steps of actually creating and finishing one. So we'll start with that P generation from our example before. T-gen. We're going to have parent 1, which is going to be homozygous dominant, and then parent 2, which is going to be homozygous recessive. Now, you can do whatever letters that you want to when you do your Punnett squares. Most people will pick one that kind of references whatever trait it is. So in this instance, we're talking about something being tall or short. So we use T for tall. I like to use the same letter for mine. However, you don't have to. You could make this second parent to indicate the short. You could do the two lowercase s's instead. I just think it's less confusing if you stick to the same letter. Now we will do our Punnett square. And we're going to pull these over. Again, there is only one allele per box or outside of each box. There's no rule to this either. You can put either parent, across the top or down the side. It's really up to you. I do prefer that you stay consistent though, so pick if you're going to do parent 1 across the top every time and parent 2 down the side, or vice versa. That way you don't confuse yourself. Now, whenever we're filling these out, we are going to pull this down and then pull this to the side. Now, again, I prefer to read mine across the top and then down the side. You could read yours down the side and across the top, just stay consistent. Now, obviously, nothing is changing here. So we're going to pull this all the way down to here. And then we're going to pull this one all the way over to here. Do the same thing for these. Pulling this one down. Now, when we do our genotypic and phenotypic ratios, this one will be fairly easy because they are all the same. They're all the same. They're all going to be this heterozygous, this capital and lowercase t. So we're going to put this as a 1-to-0 ratio. Do not put 4-to-0. That is wrong. 1. And we're going to do this 1-to-0 because ratios, if you don't know, can actually also be converted into percentages. Or into fractions. And if we do this for this one, we're just going to have 4 out of 4 for our fraction, which is going to be 1. Or if we did it as a percentage, it is 100% or 1. We're going to do the same thing for our p ratio. They're all the same. They're all going to code for tall. So our p ratio is also going to be 1-to-0. Now let's do this for our F1 generation. So to find our F2 offspring. They're both going to be heterozygous because all of our offspring were heterozygous in the last one. So we're going to pull them across the top and down the bottom, down the side. And we're going to follow the same thing. So this one will be homozygous dominant, have both of these dominant T's. And then we'll have a heterozygous, another heterozygous, and our homozygous recessive. Now, when we do our G ratios, we're going to count our different genotypes. In this instance, we have a couple of different ones. These heterozygous are the same, so we lump them together. So when we do this, we're going to have 1, 2, and 1. Now, whenever you do your G ratios, you do want to follow a specific order. So it should always be homozygous dominant, then heterozygous, and then homozygous recessive, because we want to go in order from most dominant to least dominant. Now, our P ratios, this is where people tend to get a little bit confused. Obviously, we have 3 different genotypes in total. But for our P ratios, we only care about what we can physically see. We don't actually care about the genetic makeup. Obviously, if we refer back to the lecture, this TT, this homozygous dominant, is going to code for tall. But this heterozygous, because this dominant allele overtakes this recessive lowercase t or allele, this is also going to code for tall. So we need to lump these together for our phenotypic ratio. And then we just have this one little lone homozygous recessive. And this is going to give us a P ratio of 3. To 1. We have 3 tall and 1 short. Again, you do need to keep these in ratio format. Don't just tell me that something is all tall, because their actual genotypes might be different. It is better to keep that ratio so that we can see. And again, for this G ratio, if we convert that into percentages, we would have 25% PT, 50% heterozygous, and 25% homozygous recessive, or 1-to-1. If we did this as fractions out of the 4, it would be 1 out of 4, 2 out of 4, and 1 out of 4. But your end result still needs to be in ratio form. And if it really confuses you, I do recommend writing this kind of stuff out to help you process, color code your Punnett square if you need to. That's all fine. Now, Punnett squares can be more tricky, and you will have one on your homework that is, and that's because it's going to be a dihybrid Punnett square, meaning that it is coding for two different traits. Which tends to stump students, and I completely understand why. It is a little bit overwhelming to look at. So we're going to go over the one from the homework together. And in this case, our parents are going to look like this. And R, this dominant R, is going to code for round. We're doing pea plants, by the way. Lowercase indicates wrinkled. This capital or dominant T is going to represent tall, while lowercase, again, is going to indicate short. And our Punnett square is going to be quite large in this instance. Nope, don't let me do it. That's fine. Now, I think what really stumps students on this is putting the parent alleles correctly above the top. So how we're going to do this, because we actually do not want to put each individual one across the top like we would with a single trait Punnett square. We're going to take each of our letters and combine them with the others. So we're going to pair them up. So we're going to do this by taking this first R, this orange one, and we're going to pair it with each of the T's. Then we will take that next one here and also pair it with each of the T's. And we'll do the same thing for those ones on the side. Pair up these, pair up these. It's going to look like this. This one, going down the side is a little easier because we have less variation. And then we're just going to do the same thing where we follow and pull down and from the sides to fill in our boxes. So we have this double R, both are capital or dominant. Our dominant T from up at the top, and then our recessive T from the side. Because these two are the same and these two are the same, it kind of helps fill this out a little bit quicker. You know that this is going to be the exact same for these two blocks right here. And we'll just keep filling this out. This one's going to have 2 dominant R's again, except for it's going to have 2 recessive T's. Keep filling these out. I'm going to just go through these quickly so we're not here all night. Obviously, these are going to be the same. These are going to be the same. These are going to be the same. Don't just pull them all the way down, though, because we do see that these change. So now we're going to have the capital R, a lowercase r, capital T, lowercase t. Again, always put your capitals together and keep letters of the same type together. I promise you, if you get in the habit of doing it, it will make things easier for you in the long run, especially working on dihybrid Punnett squares like these, where it can get confusing very quickly. Again, we know that these are going to be the same. We're going to have 2 recessive R's for this one. And also for this one, except mark this as the same here so no one gets confused. Anytime you see this arrow, it just means that it's going to be the same as the one above it. Now, if we do our G ratios for these, which I don't ask you to do in the homework, but I do ask you for the P ratio. And sometimes figuring out your G ratios can kind of help with that. You can see that we have these as the same. However, we also have this one. It's going to be the same as this one right here. It's going to be heterozygous for both traits. We have this where one trait is homozygous dominant and one is homozygous recessive. This one where we have a heterozygous for one trait and a homozygous recessive for the other also has doubles. And then these are each their own type. So if we were going to do our G ratio for these, it will actually end up being 2. We have 4 of these red ones. And you can count all these out like I'm doing right here before you simplify if you find that helpful. I think it just takes longer, but we'll do that for this one so that you guys can see what it's like. Again, you can do these like ratios and simplify them so that we would actually end up having you can also put these in order, so we could do simplifying these, we could turn it into 1 to 2 to 1 to 2 to 1 to 1. But then you would want to put it as 2, 2, 1, 1, 1, 1. But I'm not going to ask you for the G ratio for the dihybrid Punnett square because it just takes too long. I will ask you for the P ratio though. So as you can see, we've got our genotypes kind of separated here. And we've got this kind of homozygous dominant and then heterozygous. So we know that this is going to be round and tall. But we also have these 4 that are heterozygous for both traits. They're also going to be round and tall. So what are we going to do? We're going to lump them together. There we go. If we move on, we've got this homozygous dominant with the homozygous recessive. So it's going to be round and short. But this other one that is heterozygous and then homozygous recessive is also going to be round and short. So we're going to lump them together. Now, each of these last little boxes there, they are not going to be lumped together because they don't code for the same thing. So this homozygous recessive and then heterozygous is going to be wrinkled and tall, where this last one that is all homozygous recessive is going to be wrinkled and short. So they're by their lonesomes. So now that we kind of have these lumped together, let me erase some of this for some room. We can actually figure out our P ratio. So we have 1, 2, 3, 4, 5, and 6 of the round and tall. We'll give that a highlight so that you guys can see where it's coming from. We also have 1, 2, 3, 4, 5, and 6 of the round and short. And then we have 1, 2 of the wrinkled and tall, and 1, 2 of the wrinkled and short. Now, if you want to, you can then simplify this into 3, 3, 1, 1. But you don't have to. I'll know what you mean with the 6, 6, 2, 2, and that's perfectly fine to indicate the total number of offspring for our Punnett square in this particular instance. Now there is one other question on your guys' homework that is going to show you a Punnett square of blood types. Very often students are very confused by this. It's going to ask you about inheritance patterns. So which inheritance pattern, whether that be complete dominance, codominance, or incomplete dominance, that the blood type follows. And you should be able to see that by looking at the offspring genotypes and which blood type they end up presenting as. There will be some differences that you should be able to notice. And it is up to you to then decide how the different blood type indicates inheritance patterns. But don't tell me that it follows inheritance or the genotypic and phenotypic ratios for it. That's not what I'm asking you for in this instance. I want to know whether it follows complete dominance, codominance, or incomplete dominance, and why. But that is all I have for you guys today. So if you have any questions, please let me know. If there is a lot of confusion about some of these Punnett squares, then we can definitely discuss it more later on. But that is all I have for you guys tonight. So I will talk to you next time with next week's lectures.